the ELISA pointer. Yes, I like to thank EPNOE, the organization, very much for inviting me to give this talk about our work on the antimicrobial structure activity relationship for chitosan and the derivatives of chitosan, and the latter thing is maybe more that we are emphasizing. And as said, I'm a professor of medicinal chemistry here at the Faculty of Pharmaceutical Sciences in Iceland. And but the focus of our research has much been on the chemical modification of chitosan, and it's used as antimicrobial agent, and also in drug delivery. But the focus here will be on the antimicrobial aspects. But you know, as this might be a diverse audience, then I also like to start with making some introduction just about chitin and chitosan, and a little bit the general issues that you have to think about also with antimicrobial studies. And then I will go into our studies, a little bit summary, mostly of our studies, but also some reference to literature on the investigation of this structure-activity relationship, and then summarize with some conclusion. And I will also give reference to some of our recent reviews if you want to, you know, read more on what I'm going to present. And first, I am going to talk about chitin and chitosan. So chitin is maybe most people who are in the polysaccharide will know this. This is a linear homopolysaccharide of poly 1,4-N-acetylglucosamine, and the structure is shown here. And this one is, and it's first and foremost a structural polysaccharide. So it's found in the animal, we should say animal kingdom in general, and in the fungi. So in this site of this, you know, the kingdoms of life, probably in bacteria, but we don't have these as much investigation there. But what is most well known, it is in fungi, a part of the cell wall, but then it's a structural polysaccharide in like corals, forming the inner skeleton, and then the exoskeleton of like insects and crustaceans like crabs, and then you have it in many kinds of animals, also like octopuses, cephaloids, etc., fishes, and apparently in salamanders, but as far as we know today, it is not found in mammals, and it's not found in plants. However, it has very similar structure and properties to cellulose, and the only difference is, of course, that we have this N-acetyl group in chitin, whereas we have just hydroxyl groups in cellulose. Like cellulose, it forms very strong intramolecular hydrogen bonds, which makes it insoluble in almost all solvents, at least normal solvent system. However, even if it's found widely in nature, there are two sources for the production where we get this from, mostly from crustaceans, these shells of the crustaceans, and fungi is also maybe an increasing source. One of the things that is with like food and nutraceutical use, then if you isolate it from fungi, then you can call this a vegan product, whereas that's of course not possible for crustaceans. So chitin has rather limited use, but it's mainly used as a raw material for making chitosan, which I'm going to talk about, and also these glucosamine, and acetylglucosamine and glucosamine, which are commonly sold as nutraceuticals. So chitin, you can state that because it is actually found in some fungi, although this is not really the source. It is more produced by deacetylation of chitin, and this shows the structure of chitosan. It's actually, of course, the same as for chitin, except the amino group has been deacetylated. And now you have the possibility of proteinization. I show one here, acetyl groups, and this is most chitosan materials that you get. It always has some degree of acetylation, even if it's quite low in some cases. And it is obtained usually from crustacean shells, like here in Iceland. We have a shrimp fishing, and in the processing, the shells are peeled. This is was a waste here in former times, but now these shells are used in a processing facility where they first deproteinize the shells, demineralize them, and discoloration to obtain chitin, alpha chitin from the shells. And then in the same factory, there is the deacetylation is produced to make chitosan, which is then the main product of this. But it usually requires quite very concentrated sodium hydroxide to perform this deacetylation. Now there is what is the chitosan is important, maybe mainly because it can be polycationic, which is quite unique for like a natural polysaccharide, I would say. And as we can have proteinization of the amino groups, and if you have slightly acidic solutions, then you can actually get it into aqueous solutions, and that makes it much more applicable than, for example, chitin. Another aspect of chitosan is that it has many useful applications. In industry, these large bulk use is shown a good quality and is used in water treatment for water purification. In agriculture, there are various products. There is also growth promotion and plant protection to protect it for some pathogens. In food processing, this is, of course, it can be consumed by humans, is safe, and can be used as a preservative or flocculant in various processing industries, like for fish fillet, that you can increase the shelf life by treating them with chitosan. Then an important use of it is just as a health nutraceutical. It's sold directly to consumers, mainly as an anti-obesity product to bind fat in the intestine, but also shown to be cholesterol lowering product, although this is not approved as far as I know by medical authorities. And maybe our main interest in chitosan is due to its usefulness in pharmaceutical and biomedical application. Wound healing products are available. It's used in bone growth, and there are some products on the market or in development. There is huge literature on nanoparticles based on chitosan, used also for gene delivery. So the literature is quite extensive. There are many thousand publications appearing every year, but the the pharmaceutical use that I'm going to discuss a little bit is what is also quite unique among polysaccharide is that it has quite substantial antimicrobial effects. And this can be useful in the biomedical sector, pharmaceutical, but also in the, like for food preservation. That's also in agriculture. This is also important there. And it has quite wide properties. It's used antibacterial, and what I'm going to discuss, we have mostly discussed investigated the antibacterial use of chitosan. It is antifungal, and that's quite confirmed. Some of the problems, like Candida, there are certain fungi like Candida which causes infection where you can investigate these easily. But in many cases, the difficulty with fungal species is difficult to assay the activity. So it can be often difficult to quantify the effect on fungi, but the activity, there are many studies to show activity, and also, which is less studied, but there are a number of publications still on the antiviral properties of chitosan. So it can also used to reduce deactivate viruses. And when it's used, it's not the solid as such, of course, but more solutions of chitosan, nanoparticles, gels, and coatings. These are typical formulations that are investigated for antimicrobial activity. It's quite common to state, and I think we have done that too, that it's only active at low pH, at pH 6.5, when it becomes mostly protonized. However, we at some other groups have actually reported activity at pH 7 to 8. So it seems like that it could be also active at neutral pH. The problem is maybe more with solubility, so it's difficult sometimes to interpret the results because it's difficult to get them in the material in solution at the higher pH. So, but most investigations with chitosans are done at pH below 6.5 and not at 7. So if we talk about the antimicrobial mechanism of action, then like previously stated, it is polycationic, and that it's not some other polycationic polymers are also known to have antimicrobial effect. So this is maybe the basis for its activities, and it's thought that it's the electrostatic interaction that is important. So you have the cationic chitosan, and maybe the anionic cell surface, or it could be also bind to biomolecules through this electrostatic interaction. Early on, so this is already sometimes in the 80s, as far as I remember, then it's reported that this can disrupt cell membranes. So, and that is often cited as the activity. But since then, there are a number of studies where other activities have been claimed. Is, for example, blocking of nutrient flow, extraction of the membrane proteins to disrupt cell wall interference, binding of intracellular DNA and RNA. So this is interfering with the function of the cell, that you have an intracellular activity for small chito-oligomers that are taken into the cells, and there are various other mechanisms. And what is also very often stated is that you have a different mechanism for gram-positive and gram-negative bacteria. However, in a recent review, I was going through this literature, and my conclusion is that it's a question about all these other mechanisms and also this difference between gram-positive and gram-negative bacteria because it's even often lacks experimental verification, or there is very limited experimental studies behind it. There are a number of studies showing that there is some disruption in the cell membranes when bacteria is treated with chitosan. So that seems to be well supported by various evidence. However, other mechanisms, like here, the uptake of small chitosan oligomers have been shown, like in this study from 2008, with fluorescent molecules, fluorescently labeled chitosan. However, what is lacking, even if you can see the uptake, correlation between this uptake and activity. So the question is it really having a detrimental effect on the microorganism, even if it's taken up? It could also be just taken up as food, so not really interfering with the bacteria. So my bet is still mostly on the disruption of the cell membranes. However, this is a significant topic because even if we say that it's disrupting the cell membrane, we don't know exactly how and why this is happening. And the structure activity studies like may contribute something to help us to confirm or refute some hypotheses. I think we should also come to the question of antimicrobial assays and how this is done. What you see in many publications is that people use this so-called disk diffusion or well diffusion assay. Shown here that you place like a disk, for example, with the material here, and then you measure the activity on how much the killing zone around the disk that show the material, how much bacteria is killing. Here it's placed on agar with that has been treated with bacteria, grown bacteria. This is a common test, and maybe because it's very convenient, easy to observe. However, I would somewhat criticize this and have questions about it, which we are going to report more on later, is that this is designed for small molecules. And the size of the zone is the activity of the material multiplied by the diffusion coefficient. So small molecules can diffuse easily. However, the question is it really possible for polymers like chitosan to diffuse into the agar? And as far as we have seen from our studies, is that probably not. And this is something that the validity of such studies, at least you have to be quite careful in the interpretations. However, there is another standard method, is the so-called broth microdilution assay, where you have solution. It's quite simple. You just dilute the sample, have like 2 times dilution series, make that for your compound, and then you seed with bacteria and you observe the bacterial growth. So the And the concentration where the highest concentration where there is no growth is then the minimal inhibition concentration. Then usually it's advisable to confirm this result and validate them by plating also liquid from the wells and count the colonies. So you see also if the bacteria have been killed by this, and this is also a validation of the MIC results. So this is our preferred methods, and which we base our results, but there are various other methods that you can use, maybe not as quantitative, like live dead staining, biofilm staining, and microscopy. This also has its role in this investigation of antimicrobial activity. So yeah, when we think about chitosan, we have to think about the structure and what we have to know about the structure material that we're investigating. There are two parameters that is very important to always report is the degree of acetylation, or sometimes it's given up, it's the reverse as degree of deacetylation, because the chitosan is obtained from chitin, so then you like to say this is deacetylation. If we talk about degree of acetylation, then it's common in material from maybe 0.05, 5% acetylated up to 0.4. About 50% then, if you have more than 50% acetylation, usually you will refer to that materials rather than as chitin rather than chitosan. And it will have often start to become insoluble. Molecular weight is also important, or the degree of polymerization, usually reporting more the molecular weight for larger materials and maybe degree of polymerization for small molecules. So what we could say, what we get from different sources is less than 1 kilodalton up to around 1,000 kilodalton is the typical material that you can obtain. Some hundreds of kilodalton is maybe like a quite commonly available. So you can have short oligomers up to around 300 degree of polymerization. If you have quite low molecular weight, this is referred to as chito-oligomers or COS, and there is much interest in these type of materials as they are seen to be at least partially absorbed into the body, whereas the polymer will not be absorbed from the intestine. However, the problem with these COS studies and some reviews is that there is no universally agreed definition. So what someone calls a polymer, another researcher will call a COS, and this causes considerable confusion so people have to be careful and see what the actual molecular weight is rather than just the name that is used. The acetylation pattern is you have degree acetylation, but then you also have the acetylation pattern. Are there many? Is it just random, the acetylation, or do you have like some areas in the polymers which are acetylated and other large strings that are non-acetylated? This may not be random, especially if you use enzymatic methods for deacetylation, which I don't go into, but this acetylation is very rarely reported because it's quite challenging. However, especially with oligomer, there is a strong correlation with the acetylation pattern and biological activity, and this may also affect polymers both by both the physical-chemical properties and the biological activity. Molecular weight is not just the ideally you should also report the molecular weight distribution. These are not really available, not the larger molecules as monodisperse molecules, and you have the polymer distribution index, mole weight, that is common. And sometimes you have samples where this is rather homogeneously, you'd say, distributed, but in other samples you can have different populations and quite long tails in the distribution. So PD will not give you all the information. Preferably you should also have the molecular weight profile. But the really minimum to give information about this your materials is the degree of acetylation and the molecular weight. And then I would maybe come to this, maybe some limitations of chitosans, which should say this limited structural variation, charge is dependent on pH, there is low solubility of neutral pH, usually use, and you can get precipitation with certain acids, and low solubility in organic solvents, and that's important when you're processing chitosan. I just mentioned that purity is important, and this is something one needs to consider when doing chitosan research. There can be proteins and allergens which are allergens, and endotoxin in some products. So the difference is important when you are doing biological investigation, especially. So then I'm going to talk about chitosan derivatives and why we are doing this. This is chemical modification. We do this for obtaining chitosan derivatives and chitosan conjugates. And we can use this to improve chitosan properties, like increase or decrease the solubility and other physicochemical properties. We can use this to enhance biological properties, some activity or reduce toxicity, like the antimicrobial effect, and that's what I'm going to talk about. And when you do this modification, we have to think about the monomer, glucosamine monomer, and there are 3 here nucleophilic groups, and usually you do the modification on one or all of these groups. The primary amino group, which is under some conditions the most nucleophilic. The second most nucleophilic is this C6 hydroxyl group, and the second, then you have the secondary hydroxyl group. If you just do methylation, simple modification, then I usually call this as chitosan derivatives. But if you attach some biologically active molecule, then you can define this compound as chitosan conjugates, like linking it to protein or carbohydrate. But there is no universally agreed definition. This is just what I try to use when we discuss about the materials that we are making. So if we talk about degree of substitution, chemo and regioselectivity and characterization, then we can say when you are reporting a chitosan derivative, it's very important to report the degree of substitutions, how many substituents you have added to each monomer. And this can be vary from 0 to a maximum maybe of 5. There can be like 3 on the amino group and 1 of each on the hydroxyl group, but are often in many, when you are doing studies, often less than 1. But it's very important that this value is reported. Unfortunately, sometimes the researchers do not. And it's many reaction. It's also important to understand which group is modified, and this can be difficult to control. So chemoselectivity is N versus O modification, regioselectivity, which of these three groups is mostly modified. And the difficulty in controlling this is especially if you have higher DS, then you tend to get all the groups modified. So detailed characterization is always recommended. And this is sometimes researchers are not careful, even if you are repeating some reaction that you have done yourself or before, then the starting material can vary and even conditions. So you, for doing a reaction 2 times doesn't necessarily give the same product. So NMR is the preferred method using to determine DS and DA and the structure. And that's what we recommend, and especially should at least proton NMR 13C is very good, and then using some T2 method as a COSY and HSQC to fully confirm the structure is ideal. And mole weight determination by GGPC after you have done the modification is also good, because your process can affect the molecular weight. Problem is that many publications, they maybe use only FTIR, which is very open to interpretation, and and they are reporting not fully characterized compounds, and the question is, what is the usefulness of such finding biological activity if we don't really know what the compound is that was tested? So most commonly you use what I like to refer to as direct modification, just simple reactions with maybe one step where you like reaction with an aldehyde or ketone to make an imine, or with some acyl group to get alkylation, where you can also get alkylation, or an acylation where you can get also ester formation. So these are very common reactions. However, sometimes it's difficult to control the reaction, and in some cases the lack of selectivity can become an issue, especially when the DS is high. So therefore, what we have often preferred is to use protection group, like in a normal organic chemistry. There are 2 purposes behind this: is to improve selectivity and also to improve solubility in organic solvent. And the most commonly used is like phthalate or 3-phenylmethyl protection. First you protect with phthalate group, and then you can, for example, do modification on the C6 position without affecting the amino group. Or you can use this to introduce the trityl group here to get better solubility organic solvents, and then do some modification, which we have done. However, we have preferred to use the terbutyl dimethylsilyl protection. This is a method we introduced ourselves. It's quite simple to make the selective protection of the OH group from Chitosan here with this method, where the product separates out from the solution in 30 minutes, and you can confirm by NMR that there is full protection of the OH group, and then you can have very selective reaction on the amino group. A further advantage is that now you get a compound that is or polymer that is soluble in dichloromethane, and this makes it much easier to do organic chemistry and to get more quantitative and selective reactions. And this, we have used DCM or NMP as solvent for these reactions. We have used, done both acylations with this method or alkylations of different ways. We have a very good control by this method of the DS, but incomplete deprotection can be an issue. And also when you perform all these steps, especially in the end, when you treat with acid, then you can get a mole weight reduction in this. So you have to, so it's advisable to measure the molecular weight. And then I'm going to come into the main issue maybe of my talk, which is the structural activity relationship. And a little bit talk about why do we study structural activity relationship, especially for Chitosan derivatives. Well, this is important to maximize the activity, minimize the toxicity, and often have good solubility. In my opinion, it's also a little bit questionable when you have just one derivative made, and measure the activity. Ideally, you should make a range of activities to validate this result. The similar structures should give similar results. And inform about the mechanism of reaction to confirm or refute some hypotheses. Important structural variants are then substituent structure, the degree of substitution, and the molecular weight. If I talk about the substituent structure, this is a study, for example, that we did with antimicrobial and 3-alkyl derivatives where they are fully substituted because we use TBDMS. And there we have here 3-methyl, and then we gradually increase the length of the alkyl chain, or we have 2 longer alkyl chain. And we get a very interesting structure activity relationship. First of all, the hemolytic activity, or what we could say is toxicity, increases very significantly with the length of the alkyl chain. With the short alkyl chains, you have almost no toxicity, but this is quite significant with the longer alkyl chain. However, with S. aureus, you see that there is the activity is actually highest with relatively short alkyl chains here. However, if fecalis, which are another gram-positive bacteria, you have the opposite effect, that there is the opposite relationship, longer alkyl chain more active. This is the same for gram-negative bacteria that we tested, but the effect is much less pronounced in that effect. So the structure activity relationship can depend on the microorganism. Benzalkonium chloride, we see that these are common antimicrobial agents used in various disinfectants. They are more active, but they're also highly toxic, so to speak, or hemolytic. So the 3-methyl chitosan is actually seems to have more favorable profile, therapeutic profile, and this is also true for the antimicrobial peptides. And here is another work that we did in the synthesis of N-acyl derivatives, where we make an N-acylation and then add a cationic group, but we have different spacer length. We have the 0 carbon, 2 carbon spacer, and then 1, 2, 3, a 5 carbon spacer, separating the charge from the polymer backbone. And to confirm the result, we have 3 different starting materials, Chitosan starting materials, and in every case we do these 5 derivatives that have the same structure, and we get similar activities. And the interesting effect here is that if we increase the spatial length here, we get increased spatial length, and then we get the activity decreasing. So the activity is most highest here when we have the charge close to the polymer backbone. And then degree of substitution is another important factor. And here we have studied the antimicrobial activity and degree of substitution. This is the guanylylated Chitosan. Actually, we also studied here the spatial length, and we get the same result. The closer group is to the backbone, we get higher activity, but also the activity increases with degree of substitution, which is something that we should see if it's having a positive effect. And the maximum is around 50% degree of substitution. And after that, you do not see so much further increase. Here is a more recent study where we, again, we did with 3-methylation. We also made a tack-in, that what we called is actually more like a derivative of chitin, where we have the amino group either an acetylated or with this an acetyl 3-methylammonium. And here we get a very strong correlation. And I didn't really introduce these graphs. Note here that this is a logarithmic scale. Here you have the MIC values, and activity is up, goes up. So we see as the MIC value was lower, and we see here that with this compound, there is a very direct correlation, very strong correlation with the degree of substitution. However, with the 3-methylated derivative, where we have here 3 amino groups, then we get an increase, and then it is a plateau after around 20–30% degree of substitution. And here with 3-methylation and demethylation, we also get this increase, but then you get the same activity. But we end up with similar activity when it comes to MRSA, multi-methicillin-resistant Staphylococcus aureus. This is also another, actually we show here the same result, and here from the results I showed here, these are just shown together that you have this correlation when you have a cationic group, and this is guanidine group, which is very basic and will be cationic. There is a very good correlation between the degree of substitution and activity. However, in other cases, you can say, like here is the hydroxypropyl derivative of Chitosan. There you can see that there is this opposite effect, that with the activities are actually reducing with the degree of substitution. And this we can say propyl group is actually reducing the activity of the polymer. And that's how it's important to say, what is this substituent actually doing with regards to activity? Structural activity studies are important in this regard. And for multiple substituents, where we study solubility and activity against E. coli, S. aureus, and like a activity hemolysis, which is like a proxy for toxicity in this study. And now, when you have 3 parameters, there is actually unlimited number of possible structures. And to reduce the number of experiments, we used the design of experiment approach, where we try to make a statistical model of the results, which is like the first and second degree, and also the interaction between different parameters that we are trying to investigate. In this method, we used and we make this derivative in 1-pot synthesis to make the synthesis. Fifteen experiments. Yeah, 15 experiments. And this is shown the result, what we measure. Because we use TBDMS chitosan, we get results very close to the intended degree of substitution. But these are results of the NMR analysis. Here are the activity data, and then you use the software, you remove non-significant factor, and we see here that there is a strong positive correlation with activity against bacteria, but also with the hemolysis, with the trimethylation, the cationic charge. There is no effect, of the acetylation on the activity, whereas it has negative effect on solubility. It's kind of neutral for activity. In some regard, the lipophilic growth steroid is somewhat does not have degree effect is somewhere. So there is some synergistic effect, and we make to represent this result, we make this response contour plot. And finally, we could set some criteria for optimization. So high antimicrobial activity, high solubility, and low hemolytic toxicity. And then the proposed derivative is this, and the predicted values are as shown here. And if we take the derivative that is closest to this, then we see that the observed values are actually quite close to what is predicted from the model. But we did some statistical tests. I didn't go into it, that were used to validate this model of the activity. Finally, yes, the time is maybe going faster than I hoped. Is the molecular weight versus activity of chitosan. This has much been studied, but we reported this study here in 2019, where we studied many different derivatives. We made many different molecular weights of chitosan and 3-methyl chitosan, and we observed this interesting relationship that the activity increased very dramatically with molecular weight until we reach a certain molecular weight, which we call critical mole weight for activity. And after that, molecular weight doesn't seem to have any effect on activity. Now, in the past, there have been many studies of the relationship between molecular weight and activity of chitosan. We were kind of wondering why people are reporting very much different results from these studies. However, if we reanalyze data from previous studies, here from one from 2014 and another from 2003, we see that that actually the data is quite similar to what we have observed. There is an increase up to a certain molecular weight, and then you don't see much change in the activity. It's what researchers have maybe reported different conclusions, but the data is very similar. It seems like that there is some critical mole weight for activity around maybe 10 kilodalton, where you have the maximum activity, and then there is no further increase. But this is something that we like to study much in more detail in the future. And so, yeah, the time is up for some time. Conclusion: the antibacterial activity of chitosan and chitin depends on the structure of the substituent. The cationic substituent can contribute to activity. And I didn't go into all these results that we have, that neutral and anionic substituent generally have a negative effect. Proximity of the cationic charge to the polymer backbone is important, and the lipophilic moieties have often negative effect, but there can be some synergistic effect with charge group. And the activity of derivatives, and this is important when you are reporting new chitosan derivatives, is that you should see some kind of correlation with degree of substitution. So this is an important step to validate that where is this a good active derivative or not. And the activity will increase the mole weight until critical mole weight for activity is reached, and after that there is no further increase in molecular in activity, should this be with molecular weight. However, we need more studies to confirm this observation, but it seems like that previous studies mostly support this kind of this hypothesis. And finally, I just give here a list of reviews that we have recent that is also in somewhat the basis for this talk. And finally, I would like to thank everyone involved. The funding sources, different people I have worked with. Martha, who is now a professor emeritus, this is the microbiologist. And these are my PhD students, past PhD students and current. And Vivian is, so we'll talk now afterwards. And well, she is not professor yet, but well, I think she will be possibly